Thank you for attending today. I'm pleased to introduce Robin Martin. She's an associate professor at Arizona State University and studies both terrestrial and marine systems via remote sensing as well as in situ. So going in there and looking at the closer parameters of what she's studying. So here's Robin Martin.
Thank you, Kimberly. So, hi everyone. First of all, Greg wanted to let you know he's very sorry he can't be here today, but he has to teach class. But I'm going to talk to you a bit about the Center for Global Discovery's Airborne mapping program that was actually born here in Hilo and has operated here since 1998. And I'll talk to you about that in the context of airborne mapping for science and management and focusing on ROD. Recently, you might have seen Greg's talk about our mapping of coral reefs, and I just wanted to show you quickly some of the results. These are the live cover results we mapped all 8 Hawaiian islands, and this is just one of the results showing live cover around the islands. And the paper and data can be found at hawaiicoral.org. And this was a collaboration between DAR and NOAA and us and DLNR. But I focus mostly on terrestrial forests and their applications. And so I'm going to talk to you today about about our program, why we built the Airborne program, what it does, how it works, and some specific applications to ROD. So why did we build an Airborne program? First of all, there is a great disconnect in scale. So ground surveys only cover a small area. Drones can cover a larger area, but don't have the spectral detail needed for automatic detection to really utilize all the information of the larger area. And then satellites can image a very large area, but don't have the spatial or the spectral resolution to really be useful for management of applications. So Airborne platforms give you the best of both worlds. They have fine spatial scale over large information over large areas. What makes up the Global Airborne Observatory? It's an integrated system on this gold sensor here is a visible to shortwave infrared spectrometer, imaging spectrometer, and then the silver box to the right of it is a LiDAR system. And this is the spectrometer is a passive system that collects up sunlight reflected off the surface of the Earth, and the LiDAR is an active system that sends out laser pulses and then retrieves those back, and the time it takes to get up and back then gives you information about how far away and the Earth was from the aircraft. And the tight alignment of these instruments allows for an accurate positioning of the spectrometer data on the ground. So within the LiDAR also there's a high resolution positioning system that allows 3D accuracy of where the aircraft is relative to the ground. And this system easily covers over 100 square miles per day. And it's now in its 10th iteration. So this aircraft down here started in Hilo as a very small plane with only a LiDAR and a small spectrometer, and now we have a fully outfitted aircraft that, if you had seats in it, could normally seat 21 passengers, but it's just full of our instruments. So how does imaging spectroscopy work in our system? And as I said, it's laser guided. So the LiDAR is giving precise location of the pixels. Sorry, I need to get rid of. Okay. And the spectrometer at the same time is collecting up information off the surface of the whatever it's flying over. And so we combine the spectral information from the spectrometer that goes from all the way from 400 to 2500 nanometers at very high spectral resolution, so 1 nanometer bandwidth. And we combine this with the precise location of the pixels from the LiDAR. And that pixel size, depending on how high you're flying off the ground, can be anywhere from 10 centimeter to 2 meters. So, and the height of those pixels is also recorded, and that's what can give you the shape of the canopy. But with imaging spectroscopy, we focus on the point on the canopy which is most sunlit, so that we can have the best detections and use the chemical information. So this laser-guided imaging spectroscopy gives us both information about the canopy chemistry. Shown here, you can see different wavelengths give you information about different chemical constituents in the canopy foliage. Water is shown in blue lines and mostly is present in the near IR region, water within the leaves, and then pigments and nitrogen are in the visible portion, like when you think of chlorophyll, that has a lot of nitrogen in it. And then also out in the shortwave IR in other compounds like lignin. And then there are also carbon compounds like non-structural carbohydrates and lignin and cellulose that can all be detected using the imaging spectroscopy. It also can be used for species mapping. So this middle graph shows different spectral signatures from different species of trees. And you can see that they all have slightly different bumps and wiggles, and those can be used to map out each of the species. And this is only possible because we have the combined LiDAR and spectrometer, and that allows us to do a screening process so that we can get the fully sunlit pixels, so that we know that the measurement we're taking is actually of the chemistry and not of shade or a dead tree or something. So we can know we're getting live foliage for our measurement. And that allows us to map the species or their chemical concentrations across really large areas. This is an example of Mauna Ka'a and using the full spectral measurements. So back to ʻŌhiʻa, the topic of today. It's a keystone species and it supports critical parts of our ecosystem, and as we know, it covers a huge range across all islands in Hawaii, and from lowland all the way up to tree line. But unfortunately, we have rapid ʻŌhiʻa death, which was coined in 2010, and it's characterized by browning and asymmetrical wilting, which then is followed by tree death in weeks or a few months. And it's caused by two fungal pathogens, Ceratocystis leuco-ōhiʻa and Julio ōhiʻa. And Julio ōhiʻa is a pathogen that sort of has a slower rate of infection and death, and ʻŌhiʻa is where the pathogen that you see when you see like a whole stand that's brown. And these symptoms are really different from other ecologically related tree death, like drought, which is a more uniformly spread problem, or other types of death that might be found in ʻŌhiʻa. So it's a very unique pattern. And here's just an example from our plane, and you can see the brown trees sort of scattered throughout the landscape. So how do we detect ROD? Well, we use the spectral signatures. And so from field spectrometers, which take the same measurement, but luckily are handheld, so you can just pick the leaves that you want to measure. We found that the healthy green leaves are shown here in the green lines, and then the ROD-infected brown leaves are shown in the orange-brown line, and you can see that they have really distinct shapes. So, and these are most distinct in the shortwave regions of the spectrum, and there's in the visible 400 to 700 nanometers, you can see that there's a distinct difference where the chlorophyll has gone, which is what causes that absorption feature in the green. So using these same patterns were found when we use the aircraft data. And in areas of known ROD, so walking around in the field, the brown spectral detections aligned with confirmed ceratocystis at an 80% correlation. So using this information, we model the spectra and use this to make maps. And now we have, this is fully operational, so the models can work. We can just pull in new data and make new maps of brown trees. And this was done using training data from brown, green, and we also needed to separate out the leafless trees. And so a lot of computer modeling, and we use the filtering that I showed you before for the spectra. And in the end, these create maps that are at a subcrown resolution of 2 meters for any of the dense forests that we've flown with the aircraft. And this is image showing our data to date. So we flew in 2016, 2017, 18, and 19. And the maps here show the density of ROD in a 300 by 300 meter area. So the number of pixels that had a ROD detection, and the detections varied from year to year, ranging from about 4,000 trees or pixels to about 60,000 trees, over 60,000 trees in any given year. And the densities within the areas ranged from 5 to 200 pixels. And the outbreak patterns varied from year to year. So you can see really high density, the yellow sort of shifting around the island as the time progressed. And this is both affected by the outbreak itself, and then also the fact that you've kind of killed off areas already, so they're not going to have as many trees to have ROD. And then in 2018, you'll notice that we didn't fly the southern Puna region because it was just simply too cloudy during the time that we had the aircraft out here, so we couldn't get that area. And then we also verified these results by using a high resolution camera that we have on board. And that's the image shown here on the left, and then the right is the spectrometer data that we get that's at the 2-meter pixel resolution. And we mapped the brown ʻŌhiʻa at nearly 87%. And so very, very good detections for the brown trees. So using these data, we wanted to understand a number of factors and things about ROD, how sort of where it was, how severe the outbreaks were, and then in management contexts, and what were the sort of the environmental factors that might affect the patterns that we see. And then at a local scale, do fences help reduce the probability of brown trees? And where are the trees expanding? And then most recently, I've been working on whether or not we can use the chemical signatures as an early detection to find trees that might not have visible symptoms yet, but are sort of on their way to becoming brown, but you can't see them with your eye. So modeling the environmental influences, this is helpful because it can then show us, what the balance between like what's going to be just happening and then what we could manage for. And we used a number of input maps and again, computer modeling, artificial intelligence to to find where there were brown trees. So a presence of yes or no, and then if they were present, how many crowns were there in that area? And actually, I think this is supposed to be a presence map saying yes or no from 0 to 7. So this is the severity of numbers of pixels in the area once the presence is detected. And this is presence and absence, and the environmental factors explains 76% of the brown ʻŌhiʻa locations, but the most important factors were tree cover and height, which we got from the LiDAR, which you have to have the LiDAR data to get those that information, and then rainfall aspect and wind speed. So this is really saying that the forest characteristics, like how close together the trees are and how high they are, have a major effect on whether or not they're going to, we say brown because we don't know for sure every single tree has ROD, but in general, the ROD comes into trees through wounding. So if you have a dense tree canopy and you have high, tall trees that are up in the wind, then you're most likely to create little wounds that the ROD can get into and affect the tree. So then other factors beyond the forest characteristics that were important were weather and topography, and substrate age was also an important factor. And the substrate age often also has to do with whether or not you have ʻŌhiʻa or pohoihoi lava, because pigs don't like being on the ʻŌhiʻa lava, and so they're not rubbing up against the trees and creating wounds. So one other interesting thing we found. And then this is the map of the severity. And so the detections within an area, and environmental factors explain 34% of the brown ʻŌhiʻa tree locations. And for severity, the most important factors were rainfall, distance from roads, and cloud frequency, which is also kind of suggestive of rainfall. And these findings suggest that the hydrologic status of the trees. So wetter trees are associated with higher severity of rot and more than other environmental factors. Then we also used the maps to look at the local effect of fencing. So if you have an area, can you close it off to ungulates, and whether or not that would have an effect. So this is after you account for the environmental factors. And we used two locations on the Big Island, and it's a comparison of models at each site, so inside and outside fences. And these are the results. So this is showing the probability of brown trees outside in yellow, and then inside the fenced hashed white areas, you can see that there's a much lower probability of rot infected trees. And brown trees. And these were highly significant effects after environment was accounted for. And in both the sites, there was lower presence inside the fenced areas than outside the fenced areas. And then we also wanted to look over time to see if these maps could be used to understand rod expansion. And so using the maps made from 2016, 17, and 18, we looked at 10-hectare parcels, and so which parcels didn't have detections in them in the previous year. And the newly affected parcels are shown here. So changing from 2016 to 2017 in red, and 2017 to 2018 in yellow. And so newly infected means that there were 3 or more detections in that 10-hectare parcel that had no previous detections the year before. And you can see there's between 40 and 45,000 new rod detections in these different areas. And the pattern is also different. So early on, it was in Lower Puna and a little bit up in Kohala area, and some on the west side and a bit along the Kau coast. But then from 2017 to 18, it shifts more upland. And that could be due to the fact that, you know, the disease has already wiped out the trees in the previous area, but also has to do with the environmental factors that we looked at just a few minutes ago. And then here are some examples of the fastest expansion. So Hakalel Forest, Kuhuku, unfortunately, and Mahukona, and the mid-west side Kona coast. And these all have different, so in Kona, it could be the presence of people, but in Hakalel and Kuhuku, there are far less people there, so it has to be something else going on up there. So could we possibly detect this disease before we have these brown leaves that you can see here in picture B? And so we went back to the spectral signals that we were looking at, and then knowing that those are indicative of the chemistry of the leaves, we looked at which chemistries those were most predictive of, and green leaves, healthy leaves, generally have higher leaf mass per area, chlorophyll, and defense compounds like phenols and tannins, and non-structural carbohydrates, as well as a higher water content compared to the brown dried leaves that have higher carbon and cellulose concentrations than the green leaves. And we wanted to look to see if there was a quantifiable difference in this signal prior to visible browning, and then could this signal be predicted and modeled so that we could look at it across the landscape. And so we selected trees in 2018 here on the left that were green in 2018, but then in 2019 turned brown, and they kind of look purple here in the 2019 image. And we did this at a number of sites across the Big Island. And we did find that there were measurable differences in many photochemicals between the green and the to become brown crowns, even when you couldn't see the difference. So on the right here, the top graphs are showing like true color image and a color infrared image, which usually highlights water content. And you can see there's no real difference between the green to become brown crowns. But if you look in the canopy water content or cellulose chemical maps, you can see that there is a difference between the two. And on the left here just shows some histograms of the different chemicals concentrations in the different images for the green and brown to become brown crowns. And it looks like we can predict the to become brown crowns about 72% of the time. And we did see this similar signal in all the regions that we looked at. And so this is extremely promising given that the highly polymorphic, the different forms of ʻŌhiʻa that you find across the island, from the tall stands to the little scraggly shrubs. And but we found the signal in all of those different forests. And this really signifies that the canopies could maybe stressed long prior to showing symptoms, and that just indicates that they are susceptible to rot. So in conclusion, the airborne mapping allowed us to find individual tree detections across Hawaii Island, and just so finding those little needles in a haystack, which you can't walk out and try and count them all. The environmental factors explain 75% of the presence or absence of brown trees, but only 34% of the severity of the disease spread. And we found that fencing significantly reduced the probability of brown ʻŌhiʻa tree detections. And there was rapid expansion between 2016 and 2018 that was detectable across all parts of the Big Island. And we have the 2019 results. I just didn't have time to put them in here. But if anybody's curious, you can get in touch with us. And there is hope that early detection is possible. And this is, as always, a team effort and between funders and lots of different parts of the state of Hawaii.